I'm very excited to introduce the first RNA Collaborative seminar that's organized by the Bay Area RNA Club. So I'm a junior organizer of the BARC, and the BARC is an annual regional meeting for the RNA community in the San Francisco Bay Area, with a really strong focus on providing opportunities for junior scientists to promote their science. And here, as part of the bigger RNA Collaborative seminar series, we basically hope to convey the spirit as well, that we have been trying to help young scientists to promote their science over the last couple of years. And then I also quickly want to say, so we're going to have two speakers today. We're going to have Stephen Floor and Cong. And so they're both going to present, and then we're going to have quick question sessions. And I think in order to keep the questions a little bit organized, I'd either encourage you to either pose your question in the chat box and I can read it out, or also just maybe take a quick note in the chat box, or let me know if you have a question so that I can call out people that have a question. So yeah, of the two speakers that we have today, I'm going to give a quick introduction to Stephen Floor, who's going to be the first speaker. So Stephen has really been a member of the RNA community for many years, and has a very broad spectrum of research that he did throughout his career. So he started this PhD in the Gross lab at UCSF, where he looked at the processing dynamics of decapping, primarily looking at BCP2. He then moved on for a postdoc in the Doudna lab, where he did a really large-scale effort to map translational control of gene expression, and pinpointed primarily the 5' UTR as a main regulator of translation. Since 2017, he's an assistant professor at UCSF, and continues to study regulation of translation, and that's what he's going to be talking about today with his talk named RNA Chaperones, Remodel Gene Expression. Yeah, so with that, Stephen, it's all yours.
Good to see you, Stefan, and thank you for the kind introduction. Hi, everybody. It's great to see this fantastic turnout of RNA enthusiasts, which I sort of accidentally became by working with John Gross, and haven't quit working on RNA because I think there's just so much exciting research taking place. And I'll give an extra shout out to the Bay Area RNA Club, which is really a phenomenal conference that I think is on par with national meetings in RNA biology. And thanks again to Stefan and all the other co-organizers of the Bay Area RNA Club, which really has been a treasure of working on RNA biology in the Bay Area for the last 15 years or so. Okay, can't believe it's been that long. It's making me feel really old. Anyway, okay, so I'm going to talk today about how RNA chaperones remodel gene expression, and hopefully I'll explain what that means during the talk. So in my lab, we focus broadly on RNA biology and how cells make decisions using regulation of the level of RNA. I think what we work on mostly these days is protein synthesis, although not completely. And the multiple layers of this process are illustrated in this illustration by Christish that shows the nucleus and RNAs being produced and then exported through the various fates that they have, whether it's translation in the cytosol, translation on the ER, different localization. And we're really interested in my lab in sort of what happens to RNAs kind of after they're born. So they've been processed, they've been spliced, they go into the cell to interact and produce the product of genes. How does that process work? I wanted to start off just highlighting very quickly the UNITE initiative that the NIH has, which is about ending structural racism in biomedical research. And I wanted to point this out today, both because I think this is an exciting new initiative and because they have a request for information regarding how to end structural racism in biomedical research, April 9th. If you don't know what any of that means, you can also go to the UNITE page and you can see what it means, as well as some of the data there to support it. And I just really want to applaud the NIH for taking this on. Okay, so launching into the RNA part of the talk, I think that many of us know on this seminar that control of protein synthesis achieves tight control of gene expression in space and time. This could be illustrated in a variety of ways. I'm just showing a couple of examples here. One is through localized translation in neurons. So this is a reporter that has a 3' UTR that's specific to the BDNF gene. This causes this messenger RNA to be trafficked out into a neuronal process. And the GFP protein that's produced has a nuclear localization sequence, so it goes back to the nucleus. So if the neuron's just resting on the left here, you can see that the GFP fluorescence is mostly in the soma, in the cell body. But if you stimulate this neuron with insulin, then now you can see you get production of GFP through local protein synthesis in this process. This can also be changed in diseases of a variety of sorts, which we'll talk about some today. One example of this is through changes in the untranslated regions of the PDL1 gene. So PDL1 has a 3' UTR that's repressive for gene expression. There's structural variants in different kinds of cancer, especially blood cancers, that cause that 3' UTR to be deleted, which leads to aberrant expression of PDL1, and that leads to immune silencing. This is one of the targets of checkpoint blockade therapy. So in this mouse model on the right here, if you have a tumor that's growing in normal mice and you inject poly-IC to induce immune recognition of that tumor, you can clear the tumor relatively rapidly. But if you delete the 3' UTR of PDL1, again, this is not a change to the coding sequence, just regulatory sequence, right? Now you can no longer activate immune clearance of that tumor because they're aberrantly expressing this PDL1 immune silencing gene. So, these are just some examples of translational control. I think people on the call probably know many, many more. So what does this actually manifest as? In messenger RNAs, they're often diagrammed as very simple molecules that have a beginning and an end and an open reading frame. But many of us know that in reality, they're much more complicated than that. And there's many different regulatory elements that are inside of messenger RNAs that together have to be decoded by the translation machinery to decide when, where in a cell, and how much protein to make from this open reading frame. And so in my lab, we're really trying to understand, what are the rules that lead to this decision, and how does genetic variation and specific transacting factors decode this information. So we've been particularly interested for a while now in one transacting RNA binding protein called DDX3. This I'll refer to as an RNA chaperone because it can remodel RNA structures and RNA protein complexes. It's a member of the dead box protein family. This family uses the energy of ATP hydrolysis to eject proteins from RNA or to unwind RNA structures. These are not processive helicases. These are distributive, more like chaperones. DDX3 is known to associate with the translation machinery and regulate some certain class of messenger RNAs. And it's based on data, it's only required for a subset of messenger RNAs. So for example, older data showed from Won-Yu Tarn showed that if you knock down DDX3 substantially, and you look at S35 incorporation as a marker of total protein synthesis, you see basically no change. But there's certain regulatory sequences that seem to require DDX3 preferentially. So for example, if you take the 5' UTR of TGF-beta and you attach that onto luciferase, now you'll stimulate production of that reporter if you add recombinant DDX3 protein to a translation extract, whereas your control luciferase is not affected. So there's something about some 5' UTRs that means that they need DDX3. And we were really curious sort of, what is that and how can we figure it out? And one of the reasons we were curious, in addition to just basically understanding the way cells work, is that it's become clear that DDX3 is mutated in a variety of different diseases. So almost 10 years ago now, there were a series of papers that came out that showed that DDX3 is mutated in somewhere between 10 and 30% of cases of different subgroups of a brain tumor known as medulloblastoma. And these mutants are distributed throughout the protein, but are enriched in highly conserved amino acids. And so as a postdoc, so this is just some of the examples of where they land. As a postdoc, I purified these proteins and measured how these mutants affect the ability of DDX3 to separate RNA duplexes into single strands through this standard gel-based RNA duplex unwinding assay, and found that these mutants are between 10 and 1,000-fold decreased in their ability to unwind RNA. So these are basically inactive mutants of the protein. Bill Weiss's group at UCSF took iPS cells that are predisposed to form a medulloblastoma and injected them into mouse brains where they had either wild-type cells or cells that they transduced with wild-type DDX3 or mutant DDX3. And they found that if you add mutant DDX3 on top of a medulloblastoma predisposed background, that leads to an earlier emergence of tumors than these control situations. So this says that these mutants are not only present in the cancer, but they're actually also, functional as modifiers of, in this case, hyperactivated hedgehog signaling. So we really wanted to know, what are these perturbations to DDX3 doing to gene expression to actually cause this phenotype, right? And so this project was really spearheaded by two postdocs in my lab, Srivatsa Venkataramanan and Lorenzo Caviello, who deserve the majority of the credit for what I'll be presenting today. So the first thing we did in collaboration with Marcus Lanthaler's lab, and credit also to Emmanuel Weiler there for doing this, was to do PAR-clip to measure the binding sites of DDX3 on RNAs. And so many of you are familiar with this, but it's essentially an approach where you cross-link RNAs to protein, and then you sequence all the RNAs that are bound to the protein. And what we found was two broad things. The first thing that we found was that there's a specific cross-linking site of DDX3 to the 18S ribosomal RNA on helix 16, which is illustrated right here, based on the structure of the initiating ribosome. We think that this is specific to DDX3 because we can compare to other dead box proteins and we don't see an enrichment in this specific region of ribosomal RNA. So it's not just that the ribosomal RNA is abundant, it really seems to be that DDX3 is binding to this region. The second thing that we found was that DDX3 binds very frequently to transcript 5' UTRs. In fact, to the transcript, to most expressed genes. So if you take all PAR-clip reads and you average them across a normalized gene body through a metagene type analysis, you can see a large spike in the 5' UTR and right at the start codon, and then a big decrease in the open reading frame, and what we think to be predominantly background binding in the 3' UTR or annotation artifacts. And we think that because if we compare it to other RNA binding proteins, for example, EIF3, which is a canonical translation factor, FMR1, which binds to elongating ribosomes, or MOVE10, which is a 3' UTR associated helicase, we can see that DDX3 looks most similarly to EIF3B, although not exactly the same. We can talk about it later. So then, knowing where DDX3 binds, we were curious, how does getting rid of DDX3 or expressing a mutant of DDX3 affect gene expression, right? This is really the question that we set out to answer. So we performed ribosome profiling based on the previous data that suggested DDX3 function predominantly in translation, which there's a lot of. And then we compared the change in ribosome profiling on the y-axis here to RNA levels on the x-axis. And you can see two things that come out of this. First, there's a lot of genes that lie along the x equals y, right? So for many genes, changes to RNA levels are predictive of changes in ribosome levels, so they're not translationally controlled. But there's also this set of red genes on the top and a larger set of blue genes on the bottom that show selective changes in ribosome footprints without a corresponding change in RNA levels. Now, these could come for a variety of reasons, and so we're really kind of curious, what is giving rise to this, especially this set of genes that's translationally downregulated when we get rid of DDX3. So first we wanted to show, is this really a translation phenotype, or effect? Many of us know that changes to ribosome occupancy can occur for many reasons, including changes to initiation or elongation or other processes. And so what we did is we took a bunch of these 5' untranslated regions from this set of genes that I'm showing here, and we cloned them onto luciferase, and then we put that luciferase into translation lysate, and we found that when we get rid of DDX3 protein, translation from these reporters is substantially decreased. This is a log scale. And we repeated that same experiment in cells instead of in lysate in a different cell type, and using a different degradation approach where we have an auxin-based degron on DDX3 instead of an siRNA. And this broadly recapitulates the finding of the HEC293 siRNA lysate. So this shows both that this is not some off-target siRNA effect, and it also shows that the changes in ribosome occupancy that we see are mediated by changes to translation initiation, because we can take the 5' UTR of those genes and reproduce the same translation phenotype on an unrelated coding sequence. So for the aficionados inside the manuscript, this is the basis of us referring to this as translation efficiency. Okay, so the next thing we wanted to know is, do we see any kind of signal for specific sequences in the PAR-clip regions that are bound by DDX3? And credit really to Lorenzo Caviello for this analysis right here. So what Lorenzo did was take about 50 different PAR-clip data sets and call enrichment in clip peaks through an algorithm called Stream, which both identifies motifs and identifies an associated p-value with those motifs in the park-lit peaks. So it's not just the presence of a motif, it's how explanatory is that motif for binding. And what we can see on the left here is there's some RNA binding proteins like E11L1 that many of us may be familiar with that has a well-known binding motif and also has a pretty good p-value for binding to right here. And we see a bunch of translation factors over here on the right, like the EIF3 complex and DDX3, that while they may have some motifs that are present in those bound peaks, it really doesn't look anything like these canonical sequence-dependent RNA binding proteins over here on the left. So we think that the regulation by DDX3 is not because there's a specific RNA sequence that's being bound by the DDX3 protein, but rather it's something about the transcript itself. So what we did to try to identify what that might be about the transcript was to take about 20 different features of messenger RNAs, including the presence of a top motif, the GC content of many different things. And there's a very beautiful paper by Dominique Weil that just came out very recently that also speaks to the importance of GC content. I don't know exactly what that means, but I think it's clear. As Lorenzo would say, GC content explains all of biology, and I think he's not wrong in that. And in any case, so what we did is we fed all these different features into the random forest model, and we found that 4 really popped out as being the strongest explanatory signals of DDX3 binding. The GC content of the coding sequence, as well as the PyPMTR, the presence of this polycytosine cert motif that was described by the Ruggiero lab as a 4E dependent motif, as well as, and the strongest feature was in fact the baseline translation efficiency. And this is something we've been thinking about a lot because of a really beautiful review that came out of Rachel Green's group that describes earlier work from Harvey Lodish on how kinetically you can predict a reapportioning of translation onto away from lowly translated mRNAs through a global perturbation. And so this is something we've been thinking about quite a lot. In any case, so we believe, broadly that GC content, as well as some specific motifs, but not through DDX3 binding per se, is what requires DDX3 in mRNAs. Okay, so then, I mentioned earlier that in medulloblastoma, we see missense mutants and not depletion of DDX3. There are other diseases, which I'll talk about in a second, where we do see depletion of DDX3, and that's why we originally started with the siRNA knockdown. But we also did an experiment where we used our DDX3 degron to deplete wild-type DDX3 and then complement that depletion with either wild-type DDX3 or a specific missense mutant. And what we see on the right here, when we compare ribosome profiling and RNA-seq done in those contexts, is a very different profile than the one on the left, right? So on the right here, we see much fewer X equals Y, corresponding RNA and ribosome profiling changes in this degron-based experiment with the mutant. Instead, what we see is predominantly changes directly along the ribosome profiling axis. And so this is one of the signals, and we've seen a couple of these now, that there's a different impact from depleting DDX3 protein versus expressing a missense variant of DDX3. And what that might mean, I'd be happy to elaborate on in the questions, but it's an active area of research in the lab. Okay. So everything I just described was summarized in a preprint that we posted, which was recently accepted at Nucleic Acids Research. I'm very proud of Lorenzo and Srivatsa for all their contributions to that story, especially in the last year. And what has led us to conclude is that DDX3 associates with this initiation complex to somehow remodel RNAs with specific bound proteins or high GC content. And that if you fail to remodel those RNAs, perhaps you're initiating an upstream start codon or you're otherwise failing to recognize the start codon, and that leads to a decrease in translation from the main ORF. Okay, so to summarize this bit, we found that DDX3 regulates translation of mRNAs with specific features, likely secondary structure. We found that cancer-associated mutations inactivate DDX3, that may lead to decreased translation of tumor suppressors, and this is something that we're actively working on in the lab. And that we've tested a lot of this with reporter RNAs. Okay. I just want to touch very briefly on 2 other areas that we're working on. I promise it's going to be brief. So the first one is in intellectual disability and developmental delay. It's been discovered in the past 5 years that DDX3X is a very frequent cause of developmental delay. DDX3 is also mutated in intellectual disability, and so we've been working on this with Elliot Scher and Debbie Silver's groups. And this was described in a recent paper in Neuron, where we talked about the impact of DDX3 mutants on clinical outcomes, brain development, and RNA metabolism. And I just wanted to touch really briefly on one aspect of this, which is that this disorder is predominantly found in girls, with a ratio of about 104:3 in the cohort that's reported in that paper of females versus males. And this is surprising because DDX3X also has a Y-linked paralog, and we thought that the Y-linked paralog should also be able to complement for that. So Srivat's really led this and asked a series of questions about how this might be happening, given that there is a Y paralog. And I'll just cut to the chase very briefly. These proteins are very similar, but have differences in their termini, and that might lead to different outcomes. And so Srivat's used the same degradation system and complementing, instead of with the mutant, he complemented with DDX3X or with DDX3Y. And this is, for those of us who've looked at a lot of ribosome profiling data, this is a very small number of changes to see in an experiment like this. This is many fewer than we normally would see. And in fact, we believe that most of these genes that are upregulated, this is something we still need to formally prove, is because we had to use more of the DDX3Y plasmid than the DDX3X plasmid to perform this experiment. And so we're in the middle of confirming that right now, but I'm pretty sure that's what's happening here. And so broadly, we see essentially no changes in translation when we complement with X versus Y. And we confirmed that in a couple of different ways. I think the cleanest one is this one right here, where we used our lysate-based translation experiments to complement with either 3X or 3Y. And this set of reporters that we know to require DDX3 shows really no change in translation if we complement with 3X or 3Y, despite showing definitely a reduction in translation under the degradation of DDX3X. So, we believe that the differential tissue expression of DDX3X versus DDX3Y may be what's driving this. This is data from GTEx, where we're comparing RNA level expression across tissues. This is normalized, so DDX3Y also seems to show lower expression, but it also shows tissue selective expression, specifically in the brain. And so, one of the things that we're going down is to try to understand, to show this in actual tissue on our own, as well as to look at the X inactivation state of the DDX3X in patients. So in any case, to summarize that last part, and thanks again to Stefan, DDX3X promotes the translation of difficult mRNAs. We think that DDX3X and Y function similarly in translation, and I should say that this is also a preprint that we've posted. And we think that this sex bias in DDX3X syndrome may be due to some tissue-specific expression of DDX3 X and Y, or some unknown factor, which is always possible in biology, especially. So this is where we do the work, or at least have been trying to get back to doing the work. And I want to thank really everybody in my lab, who I'm extremely proud of for everything they've done over the last year. An older picture here, which we need to update, a slightly newer picture here, which we still need to update. And thank you also to our funding sources, you all for your time, and thanks again to Stefan. And I'm happy to take any questions.
Thank you very much, Stephen. That was a great presentation. I can probably start with one question that arose in the chat box, while other people are then more than happy to chime in with other questions. So one question that was asked was whether some of the clip footprints that you see on DDX3 on mRNAs are basically unsuccessful scanning events. So whether DDX3 is actually scanning throughout the whole mRNA and then sort of only locates to the start site once it successfully associates there.
Yeah, I mean, we could see, I guess, I'm not sure whether that's referring to the clips in the coding sequence. It certainly could be that failure to recognize the start codon could lead to DDX3 binding in the coding sequence. I do think that there's going to be an impact of scanning on DDX3X binding. I think a lot of it is driven kinetically, sort of, so how much time are you spending in a particular RNA location? And that's going to lead to changes in the efficiency of cross-linking at that site. So anything that leads to kind of a stall or a preferential occupancy, I think you're going to see an enrichment for. There's another question in the chat about whether DDX3X binds stably to target mRNAs or whether it drops off after unwinding RNA. And I think it absolutely drops off after unwinding RNA. One of the reasons we think we see a lot of cross-linking to mRNA is because my personal, you're hearing now my theoretical model, which we're trying to test now. So DDX3 has long and unstructured, or 100 amino acid tails on both sides. And my personal theory is that it's using one of those tails to interact with helix 16 of the 40S ribosome. And that positions then the helicase domain kind of near to the messenger RNA to deal with any kind of challenging structures or proteins as the ribosome is scanning. And that what we see when we perform parclip is that we trap those interactions. And what's keeping DDX3 next to the mRNA is actually the association with the 40S. Now, to test that, we need to map the region of DDX3 that's bound to the ribosomal RNA itself. And that's something that we're working on now. Okay, and there's another one in the chat about, hi Mitch, from whether there's anything special about the RNA binding site for DDX3 that indicates whether there's specificity for certain structures. And I think that sort of relates to what I was just saying in the last question, which is that I think my personal model is that the ribosomal RNA binding is there as kind of a recruitment tool, but that's something that we need to test right now. Another question in the chat from Elizabeth, thank you. Whether there's a correlation between 3' UTR secondary structure and DDX3 binding. Lorenzo would say GC content correlates with everything, and I think that's also true slightly for the 3' UTR, although the signal there in the random forest model was much less than the 5' UTR. Whether there's a correlation with the 5' UTR structure kind of depends on how you call 5' UTR structure. I think we have seen some associations there, but we also see strong associations with GC content, and those two are very difficult to decouple, so we don't know exactly what it is right now. We're taking a couple of approaches to try to do that, both reductionist approaches where we're building 5' UTRs kind of from scratch, and also random approaches where we're taking the DDX3 sensitive UTRs and mutating them to try to understand sort of what the required features are. Stefan, should I take one more, or do you want to cut me off?
No, go for one more.
Okay, one more from Ahmed. Thank you for the question. Does the length of the 5' UTR affect the success rate of DDX3 scanning? If so, how is that happening? So I think in general, one of the challenging things has been that it seems like the lowly translated mRNAs are the ones that require DDX3 for translation. And scanning becomes less efficient as the length of the 5' UTR alone increases independent specific regulatory content. And so, I would say don't have the assays in my lab to look at that through the kind of kinetic and single molecule type of detail that you might really want to answer that question. But I think it's certainly possible that something like length could play a big role. Okay, and I think I should probably stop.
Yeah, let's thank you very much for all the questions. So with this, we already get to our second speaker for today. So these seminars are really quick. So the second speaker today is Cong, and I'll give you just a very brief overview of his work. So he started his scientific career working on genome engineering in tail end effectors, actually, at the very start, I think, of his PhD. Before then, really his absolutely phenomenal and seminal work that laid the basis of CRISPR genome engineering in mammalian cells in the Sang lab at the Broad Institute. And during his later postdoc in Aviv Regev's lab, he expanded this tool set of CRISPR applications even more, and, for example, facilitated epigenetic editing using CRISPR, and then also started developing a lot of single cell methods to study cancer immunology. And I think these are the two sort of fairly broad topics that he still continues now to work on in his own lab since 2018. When he was assigned an assistant professor at Stanford University, and I'll let him introduce his presentation for today himself, and therefore I'll leave the floor up to you. Please.
Great, thank you so much for the invitation, Stefan, and also I'm really since moving to the Bay Area, I've been really fascinated by the incredible community at the BARC, the RNA Club here. And I'm a big fan of Stefan's work. So to differentiate a little bit from our other speaker, I decided to focus more on the gene editing technology part today. We do have I was thinking about talking about another story as Stefan introduced. We also have sort of another set of work on single cell technology, and where we used CRISPR barcoding to understand transcription regulation in the context of cancer. And we're probably, hopefully going to be able to post the preprints next week for that work. So I thought today I would focus more on the gene editing part and sort of complement to Dr. Ford's talk. And to briefly introduce our group, so I actually was studying electronic engineering before I moved to biology studies. So I always thought about biosystem as a really nice sort of technology driven area for our group, where we can really build interesting and useful genomic technologies. And we have been thinking about this from sort of the input and output, like a circuit in electronic engineering, where we can really use gene editing tools and also use single cell tracking and phenotypic functional genomics to understand basically the role of cell behavior. And we're particularly interested in cancer immunology, which extends from my previous work with Aviv, but also we recently have been more started to work with neuroimmunology models, for example, with Aaron Giller here at Stanford. And today I wanted to focus a little, share one story that we recently have published on sort of a new generation or at least new type of gene editing tools based on phage proteins for RNA-guided recombination. And the motivation for us to think about, keep developing gene editing tool, even though, as most of you know, CRISPR has been really incredibly, powerful and efficient, is really motivated by the fact that most of the existing, previous generations sort of tools are really focusing on the initial or initiation steps of gene editing, where we introduce DNA modifications at desirable genomic location. And endogenous DNA repair really are responsible for repair of the breaks or nicking and perform gene editing. And this really had limited the efficiency and accuracy of gene editing. And that's why more recently, a lot of recent work really has been focusing on using new generations of editors that aims to take control of the DNA repair process, as exemplified by base editors and prime editors. And these editors are really efficient and simple. And we saw there's still limitations for the type of work we're interested in. For example, when we study structure variations or wanted to perform knock-in of therapeutic or sort of tracking cassette in cell-based models or even in vivo models, we wanted to use gene editing tools for long sequence engineering. And here, base editing and prime editing are usually a little limited. At the same time, because these editors still requires at least a nicking of the DNA within the cell, there's still sort of noticeable levels, even though controllable, but still noticeable levels of errors or off-target effects. And because of the delivery of in vivo model is limited a lot of times by size, these base editor and parameters also are still in the process of being able to deliver using viral vectors, for example, into in vivo models. So we thought, why don't we take a look at how bacteria and microbes perform gene editing? So CRISPR was a microbial tool that really was aimed at destroy, right? That's really pioneering work from Jennifer Emanuel, a lot of other CRISPR scientists that elucidated the sort of the mechanism of these sort of bacterial defense systems. And what we're trying to do for long sequence engineering or genome integration is not to destroy the target, but really to integrate, hopefully without any scars or arrows into the genome. And phage actually have evolved a very interesting system called Recombination, using only 2 enzymes to scarlessly integrate its own genome into bacteria. And this is performed by almost every single phage that we know, exemplified, for example, by lambda phage. And in this phage sort of integration system, is fairly simple as shown on the left. You have a very simple exonuclease that generates sort of a homologous overhang, and then a a very interesting protein called single-stranded leading protein, or sometimes called beta protein for lambda phage, actually can integrate through the homologous recombination mechanism based on the homologous sequence to basically integrate the phage into bacterial genome. And most phage actually only need one enzyme, which is the second protein called beta. And when we perform metagenomic analysis across over 10,000 different phage through the metagenomic database, we realize that actually over 60% of the phage only have sort of SSAP enzyme in their genome. And RACT here is actually a broad family of SSAP that's similar to lambda beta. So basically what we thought is, can we use this integration system to really perform gene editing and hopefully be able to achieve long sequence engineering without a lot of the deleterious errors where when we perform gene cutting would generate. So in a recent work, we reported that we can successfully harness this phage single-stranded leading protein called SSAP with a Cas9 gene editor for very efficient knock-in or recombination-based gene editing in human cells. And one of the interesting sort of observation we have is definitely similar to CRISPR system. If we look at different phage proteins with the same family of same activity called SSAP, they have very differential activity if we use them for human gene editing. And we identified the RACT as a major phage protein that can be very efficient in performing gene editing in human cells. And here we're measuring their activity for a 1 kilobase knock-in. And only when the knock-in is in frame, we can detect a fluorescent protein expression from a in-frame sort of knock-in of endogenous transcription locus. So this way we can understand how precision, or at least to some extent, how precision these editing events could be in human cells. And so without going into detail, we performed a lot of different sort of protein engineering to optimize the system. We're able to observe basically that using the optimal design, we can achieve roughly 5 to 10-fold improvement of precision knock-in efficiency compared to the Cas9 control. And shown here on this sort of engineering summary. And what we observe is that this particular sort of SSAP phage protein actually are quite universally robust in terms of its activity. As you can see here, across, when we compare this SSP-based knock-in editing with other published tools, it actually not only improved over some of the published editors for kilobase knock-in, but also it can synergize with existing tools. For example, if you notice, there's a chemical way that people have been using to improve CRISPR knock-in efficiency called nocodazole. And when you use nocodazole, of course, you can improve over the Cas9 control. But if you add on top this phage SSP protein, you could actually even further improve the efficiency of the system. And across the different cell types we tested, it seemed to have robust activity. And as last piece of this published work, we decided to see if it can work with different formats of Cas9. And one of the things we tested in this recent work was Cas9 nickase, because we imagine as long as you can generate a potential sort of accessible single-strand DNA, using Cas9 nicking, similar to the base editor, maybe we can still perform SSP-based recombination. And indeed, even though Cas9 nickase usually have lower activity compared to the wild-type Cas9, but when we added the SSAP protein into the system, we do observe a very significant improvement of efficiency for these type of precision knock-in assay. And we also, of course, wondered if these nicking-based editors would be able to reduce off-target effect. And you can see on the right, as we perform this genome-wide off-target assay using GuideSeq, we could observe a very significant reduction of off-target editing, including a very well-known off-target sites basically were gone after we used the nickase version of these SSP-based editor. So follow this work, what I would like to focus a little more time on today is our work on actually generating a or hopefully showing you that we can even use non-cleavage CRISPR system to support this type of recombination, meaning these phage SSAPs could perform gene editing with fully deactivated Cas9 without any introduction of double-stranded breaks or single-strand nicks. So here is what we thought would be a possible model. As I mentioned earlier, we thought single-strand accessibility would be a key for this SSAP phage proteins to perform gene editing. And a very unique feature of CRISPR is through the R loop formation of the guide RNA invading the target. It actually releases, and thanks to a lot of really great work from from Jennifer and a lot of other people in the CRISPR field, we realize that the non-targeting strand of the genome, which show on the top, actually might become accessible single-strand DNA, even though it's not really cleaved. When we use dCas9, it is still able to release this non-sort of targeting strand while this R loop formation through the RNA invasion. And what we thought we should test is if this release the single strand of target DNA might allow the SSAP to come in and perform gene editing. The goal is basically to generate something like a programmable CRE, but without the need for LASP, but instead using RNA-guided sort of targeting of these SSAP for gene editing. And we basically realized that the previous design may not necessarily be the best. So we screened through various different metagenomic SSAP enzymes from phage databases. And one of them performed significantly better than than the other ones. So we picked this particular SSAP called ECRACT for further development using this dCas9-based cleavage-free editing system. And when we really measure, as I explained, the on-target errors, and we were very happy to observe that basically compared to Cas9-based editing systems, and including actually compared to what we previously published using the Cas9-based sort of SSAP, the dCas9-based SSAP shown on the right side actually generates zero indel above the background. We think there's still possibility for minor DNA damage, but as you can see, the level of indel formation or indel errors were really minimal when we use deep sequencing, to examine the site of knock-in. And then we also performed on and off target assay. So without going into detail, but you can see a summary on the right where we compared this dCas9 editor with Cas9 HDR and Cas9 MMEG. MMEG is a microhomology-mediated editing tool that have better efficiency, but usually more significant off-target. And you can observe that if we look at what are the percentage of editing that have no on-target errors, SSP definitely outperforms the other two comparative editors. And if we summarize the different type of errors, so on the left is the knock-in for precision knock-in efficiency. On the right is the errors that we observed from different types of assay. And you can broadly measure basically how good these type of editor are compared to classic Cas9 HDR and MMEG editors. And we observed that consistently this dCas9-based gene editing tool outperforms existing Cas9-based tools because it doesn't generate a double-stranded break of single-strand nicks. And we wanted to see if this is just lucky at a couple of different small couple locations, but we definitely wanted to see how robust this is, and through measuring knock-in efficiencies at up to 8 different locus, we were able to observe very consistent performance of this dCas9-based editing system. And even in more challenging cases where human stem cells is known to be a little difficult to deliver, and Cas9 usually achieves lower editing efficiency in these stem cell-based systems, we were able to observe comparable, slightly higher efficiency using this dCas9 editing tool. And this is very comforting because it's not specific, for example, to a specific locus or specific cell type that we use. And one of the interesting idea is that if the model that we proposed earlier were to be true, that the dCas9 really just releases a accessible single-strand DNA that SSAP can access and perform editing, then we expect maybe if we inhibit endogenous DNA repair, these dCas9 editors may not be as much inhibited compared to classic Cas9-based editing. As you can see here, we try to use two different inhibitors of classic HDR pathway, an inhibitor of the mRNA complex, which is basically one of the first steps of double-stranded break and or HDR recognition and initiation of HDR repair, and then also RAS51, which is the core enzyme for HDR repair. And we observed that basically, if we use these inhibitors when we perform Cas9-based 1 kilo-based editing, Cas9 definitely had a significant drop of efficiency as shown at these two different locus. And the dCas9 SSP editor were minimally affected by these inhibitors, supporting that, at least to some extent, the SSAP actually can be independent from endogenous repair. And that is why, even with the dCas9-based editor, it could support gene editing in human cells. So to summarize a little bit of what we've found so far, we really have, through a series of engineering, to kind of try to implement a phage-based integration system using a protein called SSAP for gene editing. And this particularly were geared towards large-scale, long-sequence engineering in human cells and try to minimize errors in this type of editing, which were very prominent when we use classic Cas9-based editing systems. And because of the unique activity of these SSAP, we observed that even with dCas9-based RNA-guided targeting, these SSAP could support gene editing activity across a variety of different locus in human cells. And one benefit of the system is that it's really compact. So more recently, in collaboration with Monty Winslow, we have been able to package these systems into adeno-associated viruses. So we can use them in a very relatively standard, like 2 AAV design, 1 AAV for the entire CRISPR component and the other AAV delivering the repair template. And then we can basically use the systems in a convenient, relatively convenient AAV format for editing. And we're testing these in challenging situations like in vivo editing with Winslow and other people as well. So to summarize a little bit what we found so far, we believe that so CRISPR is a very powerful RNA-guided technology, and the unique molecular mechanism of CRISPR actually provided opportunities as exemplified by base editing tools and prime editing. And for larger scale engineering, phage recombination enzymes can be very readily scaled up to human cell application when we use dCas9-based genomic engineering. And this dCas9 SSB, I think, will be a very sort of helpful potential tool when people perform long sequence genome editing, because the existing, some of the existing editors were more limited in terms of how much people can use these tools for gene editing in human cells. And this work was really enabled by a lot of the initial members of my group, particularly CK Wang, a postdoc in the lab, Nick, and Jason, 2 students in the lab, and also Andy Fire has been really helpful in terms of providing guidance in this collaborative work, as well as Monty Winslow, as I mentioned. So we are keep developing these tools, and we hope that to take today's opportunity to share some of the development of our technology, and really really grateful for the opportunity to share the work, and grateful to my lab and our funding agencies as well. And also, I wanted to note that I saw, and thanks to today's platform, I saw a lot of old friends, Junjie, Charles, and Yue in the group. So it's really great to see a lot of old friends. Maybe I missed some of them in the channel, but it's really great to see people from Boston here. Thank you for the opportunity. Thank you, Stephen.
Thank you very much for that. Again, very, very great presentation. That was really interesting. I can start off with a quick question. So what do you hypothesize? Why are you more accurately editing with the dCas9 embodiment? whereas, I mean, Cas9 binding is more promiscuous, like has a lower sequence requirement than actual Cas9 cutting.
Right.
Well, just looking at that, you would, I mean, I would have hypothesized that that's going to be less accurate. How do you think?
That's a great question. Yeah, so we think even though, so this is actually supported by some of the biochemical work on Cas9 binding. As you noted, there is a very, very big concern, for example, that Cas9 binding can happen in a variety of different places. But actually, because the requirement of having that single strand release on the non-targeting strand through the R-loop invasion, we some of the biophysical work, like including single molecular imaging work, definitely observed that if there's not a perfect match, even though Cas9 can bind to it, how well the sequence matches the target really affects the kinetics of how well the binding is stable. And we actually, so one sort of relevant thing is we actually did a genome-wide insertion assay using next generation sequencing. And we observed that, Cas9 knock-in actually has a lot of off-target insertion, but we didn't observe any significant insertion of Cas9 SSAP, I think because to allow the SSAP to really access the genome strand for initiating that process of editing really requires a relatively stable binding of Cas9 to the target. So that requirement actually facilitated that instead of like sometimes, when people use Cas9 for gene activation repression, maybe there's some off-target binding. In this case, as far as we've observed and assayed in this genome-wide assay, it does seem that it is a very stringent requirement for a full match between the Cas9 and the target in order for the assay to actually carry out the editing events. So without this is related to how much the SAP really requires a stable formation of the complex. And imperfect matched guide RNA, for example, wouldn't support that stable complex and release of the strand. Yeah, thank you. That's a great question.
Okay. So I can go to some of the questions from the chat box. So there's one from Ellen Schrock, who was asking, she had two questions. Her first question was how large a payload you can integrate using the Cas9 SSAP. So
yes, we may have some data. Oh, maybe I forgot. Yeah, we actually tried. So we tried up to 4 kilobase of sequence insertion in our current study, and we observed that even though larger payload will reduce, for example, the overall efficiency to some extent. Sorry, I forgot to bring the figures here, but we did observe that even though here I'm showing a roughly 1 kilobase payload, we tried as long as 4 kilobase payload. Actually, we tried to insert a whole Cas9 into the genomic locus, and that was very reasonably efficiently supported. But definitely there is a sort of gradual drop in efficiency as the payload becomes larger. So that's sort of reasonable to us, but we definitely think there's hopefully room for improvements.
Mm-hmm. Cool, thank you. There's a question. Another question is, where do you see this tool fitting into the suite of CRISPR tools that exist?
Yeah, thanks, Hannah, for the question. So without, this is a particular sort of helpful tool when we think about large scale gene editing, and particularly if you're linking into, for example, if you have 2 different mutations. So one of the collaborative work we have ongoing is that, for example, people wanted to install cis-regulatory mutations across, for example, a stretch of DNA, and that was usually sort of done through either 2-step things, or even sometimes if you use base editor, you can have problems with whether there's a proper guide RNA for you to do that base editing or prime editing. So for our sort of SAP-based technology to do this type of extensive sequencing during either multiple mutations or insertion of a tag, and we have a collaborative work on insertion, therapeutic genes for gene therapy in albumin locus in the liver, and these type of tasks, I think this tool would have significant benefits being one, generally speaking, more efficient than many of the Cas9 technology, and two, definitely if you combine the editing efficiency and also the error rate to generate, how many, say you edit 100 cells, how many cells actually have your desirable editing events, I think this tool will be significantly higher yield than many of the existing tools. And this would particularly, I think, be relevant for larger scale sequence engineering, but not, for example, if we just wanted to get one SNP edited, I think this may not be the best. Like, I think base editing definitely is probably the best way to go. But interesting, just one last comment is that we have a collaborator who actually were working with Beam Therapeutics on doing base editing, but there was no guide RNA available for them to do it. So now they actually are using our SSB tool for that, even though it's a very short sequence modification because of guide RNA restriction. I think there might still be some space for this type of SSAP editing because the mechanism fundamentally is recombination, but not, for example, like a point sort of editing. Thank you.
There was one more question on how the SSAP recognizes the template DNA.
Oh yeah, thank you. I saw that. I think that's from, is this from Alan? I think how precisely can control where the payload integrates within the guide RNA target set. Yes, so I may not, actually, so the way we can control the insertion site is pretty straightforward. It's very similar to when you provide a regular sort of Cas9 HTR template. We just basically have the fully sort of full desirable sequence in the template DNA that we supply. And we use double-stranded DNA here. We have some data that double-stranded DNA is more efficient than single-stranded DNA. But basically, you can just essentially have homology arms flanking where you want to insert, and insert that, basically have that exact sequence you want in the template, and then SSB would integrate, because SSB basically first binds to the homology part, and then try to integrate that sequence. So this way, we could control precisely the integration sites. And we did actually some Sanger sequencing to verify that it's precise. So that's something we've been able to, we've been to verify. Thank you.
Great. Yeah, so the other question that was still around was how the SSAP recognizes the template DNA. So how the template associates to the recombinant.
Oh yeah. Yeah, so this actually is, I think is actually a very interesting biological question that the field of SSAP study has been slightly, shedding some interesting light on, which is SSAP needs to form, I think, at least a dimer in order to have high affinity, and that's why some of you may have noticed. I think Ethan have noticed that we use the MS2-based aptamer design. This allows us actually to recruit at least two copies of the SSAP within the same genomic, using the same guide RNA. And we think that's really critical for the recruitment of both SSAP, basically more than one copy of the SSAP to the dCas9, and also for the dimerized SSAP to efficiently bind to the template DNA. And this actually also means, by the way, that if we simply overexpress SSAP, it doesn't actually have, we have that control in the paper and didn't really observe any effect, because I think it really needs that local dimerization that's enabled by the aptamer design using the MS2 system, as Junjie and Ethan have mentioned.
Great, that was very efficient, answering the last two questions at once.
Thank you.
Thank you very much for again, mostly the two speakers, Stephen and Nate. Thank you for taking the time to present here, and obviously the audience to attend the meeting and all the great questions. I think that was a really, really interesting seminar, lots of interactions, and I'm looking forward for other seminars of the series, and then obviously also for future seminars organized by the Bay Area RNA Club. Thank you, everyone, and have a great day.